In this video, we will discuss about a universal base edited CD7-CAR-T cells that can be used to treat different patients that are affected with uh, cancers such as leukemia cancers. So we will first start with the design, how they developed their CD7-CAR-T cells and their program by which they have enrolled the patient for the clinical trials. So they have started with these allogenic T cells, as their idea is to make off the shelf the CD7 positive CAR T cells that can be used with the patients who are affected with this tumors which has the CD7 antigen. They take the T cells from the healthy volunteer donor that has this CD52, TCRαβ, CD7, and thereafter they base edited these three antigens to get rid of these three expression of these three antigens. So they use the CRISPR-Cas9 system to target these antigens so that they will not be expressed. That can be seen here, the termination of CD52, TCRαβ and CD7. And the reason why they did that is that CD52 and TCRαβ is might create the immunogenic reactions, or more commonly it is known as GVHD. It is Graft Vs Host uh, Disease, and this will also reduce the lifetime of these modified CAR T cells within the host. So it will not have the good efficiencies, as it will have a less lifetime within the host. And CD7 is reduced to avoid this fratricide. Fratricide is that to avoid the self-targeting of these CAR T cells. Suppose the CAR T cells has CD7 and it antigens can also detect CD7. So it will target one of its own CAR T cells, or sometimes it can also do this cis interactions and target its own CAR T cells, or target its own receptors on the cell surface. So once we have these CAR T cells, or the researchers have these T cells which is devoid of CD52, TCRαβ, CD7, they did the lentivirus transduction of these T cells that can express these CAR7 antigens. So this is they commonly known as they commonly call it as BCAR7, that is base edited CAR7 T cells, and thereafter they infuse this to the multiple recipients, and the timeline on recipients are mainly kids less than 16 years of age. However, they have also used this CD7 CAR T cells on the adults based on their consent. So how they did their experiment is that first they screen the patient and then treat them with this B CAR T7s at day 0, and before that they remove this host immunity using uh, lymphodepletions. And once they achieve that, they 7 days after that they infuse these BE CAR T cells, so that the BE CAR T cells has less immunogenic response. Even though these two are removed, there is also a possibility that it will get immunogenic response from other immune cells. So in order to be sure, they remove the host immunity, and then they introduce this BE CAR T7 and then expose them for this 28 days, and thereafter they assess this bone marrow to check the conditions whether they will have more tumors or not. And after that they conditions this bone marrow and remove these BE CAR T7s, because if BE CAR7 T cells are present, then it will continue to target even the T cells that has the CAR7. So this will prevent the rebuilding of the immune systems, or rebuilding of the immunity within the host. So they need to remove these BE CAR T7s after that, and then they did the allogenic stem cell transplantations, and so to rebuild this immunity. This is how they planned their treatment. This is the timeline of the treatment. And how they did the enrollment: they first screen the children's whether and they are able to recruit 9 children from the United Kingdoms and Ireland, and 2 were from Europe. And they also recruited 2 adults after their compassionate use special license. And 2 persons were excluded because they have some active infections, and 1 does not meet the eligibility criteria out of this 13. And then those, so finally they have used 9 children and 2 adults, and 2 children were withdrawn to receive palliative care for this 28 days. So 9 patients were in remissions at day 28 and proceed to stem cell transplantations. And thereafter 2 children withdrawn. 1 had transplanted related death, and 1 received palliative care for CD7 relapse, as the CD7 T cells needs to be re-infused into the patients. And then 7 were in ongoing remissions after stem cell transplantation. So currently the 7 patients has gone through this process and are under this ongoing remissions after stem cell transplantation, or meaning they are in touch to check for this follow-up post transplantations feedback. So this is how they did the clinical studies about this universal base edited CD7-CAR-T cells, which can be used in the as a off-the-shelf CD7 CAR-T cells. So this is what they have presented in their first figure. We will discuss the subsequent results in the upcoming videos. So that's all for this video. If you need to understand more about these clinical studies in detail, you can go through the reference that has been provided in the description. Thank you for your time.